The Protein Data Bank provides worldwide repository of information about the 3D structures of large biological molecules, including proteins and nucleic acids that found in all organisms. In order to understand how those molecules work, it is important to understand its shape. Such knowledge will help to know the structure's role in human health and disease, and also in drug development. The journal article of neuraminidase of A. Brevigmission, 1918 H1N1 strain by Zhu, Xu, Dweck, Stephen and Wilson, 2008. It discussed on structural characterization of 1918 influenza virus H1N1 neuraminidase. The 1918 influenza virus H1N1 was resurrected for further investigation of its sequence, functional and structural characteristics of its neuraminidase. This report provides crystallographic analysis of neuraminidase of the virus for structure-based drug design. Designing drug that's targeting neuraminidase is important since inhibition of neuraminidase can inhibit the progression of influenza virus infection. Hence, to understand the molecular structure of neuraminidase, crystal structure should first be determined for further crystallographic analysis of macromolecule of neuraminidase H1N1. The methodology of the research were: firstly, ectodomain of neuraminidase gene from 1918 influenza virus H1N1 were cloned and expressed using baculovirus expression system. Constructed plasmid was then co-transfected into insect SF9 cells for viral amplification. Thirdly, protein was further expressed by using Hi5 cells. Then, soluble 18 neuraminidase was obtained by metal affinity chromatography. The fifth method was cleavage and purification of soluble 18 neuraminidase by size exclusion chromatography. Lastly, 18 neuraminidase was then becrystallized using sitting drop vapor diffusion method. Crystallization of 18 neuraminidase is important to produce protein crystal that can provide a diffraction pattern when exposed to X-rays in X-ray crystallography. Then, diffraction patterns produced can be analyzed to determine the tertiary, or quaternary structure. From quaternary structure, the secondary structure and further protein sequence can be determined. Emboss-back transect tool was used for further determination of nucleotide sequences of 1918 influenza virus H1N1 neuraminidase. This tool functions as back translator of a protein sequence. It makes a best estimate of the likely nucleic acid sequence it could have come from, by using a codon frequency table. In conclusion, the use of bioinformatics in biological research helps in advancing the progress of new discovery and also new knowledge, especially on therapeutic fields.